again, it's sometimes it just helps to know who's around you. So we, and it's a set of circumstances that sort of led to this Eureka moment. When I came to Duke, Bernard Amos, who was my division chief and who was just an amazing person and scientist, was not much of an administrator, you know. So I arrived and there was nothing ready for me, no lab, you know, sort of. He literally walked me to the professor in the department who had most space and said, "You think you can give her something, you know, a bench?" It was really quite amusing at the time. But I trusted him, you know, it was okay, so I'm here. And it happened to be in the lab of Dr. Richard Metzger, who was working on generating monoclonal antibodies to human tumors, tumor antigens, to see, you know, we still do that, to find immunotherapeutic targets or therapeutic targets. And it was also, you know, very shortly after the monoclonal antibodies became a technique. So he was immunizing mice with various human tumors and generating antibodies. And he had, once they had the antibodies, then he had students trying to identify the molecules. Well, I had done a lot of the kind of, you know, biochemical molecular studies at Stanford that really would help them identify this. So in exchange for space, I was overseeing his students' experiments. And one student, who was a very talented student, and I, there was an antibody that they could never identify what it was seeing. Clearly it was staining tumors, but on a gel, you know, which is what, you know, we all use to identify at least the molecular weight of the. We would make precipitates, we'd run it, nothing, you know, there was just blank gels. And, you know, then I was working with the student and I said, let's keep, you know, the top of the gel intact, because usually the top where you load the molecule, you cut it off, you throw it out, you expose the gel to film that is radioactive. So I realized that whatever we were trying to resolve on the gel was just staying right where we loaded it. And I said, it's just too big. You know, whatever it is, it's huge. So let's run it on, not on a regular protein gel, but let's run it on a DNA gel that allows huge molecules like DNA to go through. And so we identified this molecule, and it happened to be MAC1. I thought that I had not necessarily wasted my time, but I was basically paying rent, you know, by advising this student. Never occurred to me that it would be a lifelong interest until independent, you know, in my own lab, independent of Dick Metzger's students or anything, we were growing, you know, a couple of years later, several years later, we were growing human T cells from lymph nodes from cancer patients that drained the tumor site. And we were asking, what do the T cells that are expanded in the lymph nodes see on the tumor? Because the T cells are the ones that are going to kill the tumor. And we discovered that the T cells we were growing for many patients, those for pancreatic cancer patients, were killing tumor beautifully, but all tumors, I didn't really care about the HLA, you know, so my important molecule, they didn't seem to care about that. So they were HLA unrestricted. So we tried to sort of draw a picture of what would a molecule have to be like to have that ability to be recognized by the T cells without any participation of HLA. So we proposed a really sort of crazy idea, which is not that crazy at all, actually, now, and that is that there would be a molecule that would have sort of repeating units so that it would cross-link T cell receptors, and those units would be very stable and look the same from one to the other. Cross-link the receptors, activate the T cells, and the T cell will kill the tumor. So we had this picture of what the molecule would be like. Simultaneously, in the biochemistry department, the head of the biochemistry department, Bob Hill, was working on glycosylation of molecules, all in glycosylation. And he was using a pig mucin as a model molecule that is very heavily glycosylated. And so if you want to study glycosylation, you might as well take something that has hundreds of sugars on it. But he was also interested in where those sugars are, so they actually cloned the gene for the pig mucin. And I heard about it, I heard a seminar. And the gene for the pig mucin had what is now known as the typical mucin region, tandem repeat region. So it had 200 and some amino acids in a repeated 200 sometimes. And those amino acids, many of them were scaffolds for sugars. So I looked at that and I thought, that's really interesting, because that would be exactly the kind of molecule that could cross-link the T cell receptor, just the tandem repeat region. And then I said, we have this molecule on the tumor cells from this antibody that we identified, this big molecule, So that was the Eureka moment. I thought, you know what? I'm sure that it's the mucin. And sure enough, we used the same antibodies that helped us identify the molecule that Dick Metzger was interested in to block the T cells from seeing the tumor, because once the antibody would bind to the molecule, to those same parts where the T cell would bind, the T cells couldn't do it anymore. They would not kill the tumor. So we showed that actually the same thing seen by the antibody was being seen by the T cells and the T cell receptor, whereas other T cells, grown to some other antigens, we couldn't block with that antibody, et cetera, et cetera. And that's what the PNAS paper is. But it's one of these, and it was completely unexpected. The T cells were not supposed to bind native proteins. T cells were not supposed to be HLA unrestricted, none of that. It was an aha moment and a scary moment too, because, I knew we would have a hard time publishing that. We would have a hard time getting funded, because people go, what? But to the credit of all my colleagues, and I was very young, and who was I, right? And I wasn't publishing under, Dr. Kaplan or Dr. Levy, but to the credit of the community, we published a lot and we got funded and a lot, and pursued this. And now everybody knows that's what it is, and we grew other cells that were MHC restricted, HLA restricted, etc. We have vaccines all from that tandem repeat region. That was so, but it was the first time that we had seen the molecule of that sort.